Good morning, everyone. I'm Chad Laughlin with the San Diego Regional Board, so I'll be moderating the first session, just coming up right now. And this is from the San Diego Regional Board's neighbor to the north, Region 8. And it's going to be Raj. He goes by Raj. And he's going to talk a little bit today about how some little organisms can do some big things.
Thank you, sir. Yes, as Chad mentioned, my name is Gaurav Rajen. I go by Raj. I work as a water resource control engineer with the Municipal Stormwater Unit in the Santa Ana Regional Water Quality Control Board. I'm going to talk to you about Archaea, their role in methane emissions and climate change, and also maybe a little bit of an aside, how they might help in carbon sequestration, maybe converting CO2 to fuels. But I want to say, before I joined the water boards, I used to work in academia and also at a DOE national lab. So I remember going to presentations where, the speaker covers the board in slides which have a lot of mathematics, and the chair says, any questions, there's usually an embarrassed silence because no one's quite understood what's been presented. So in those cases, there are standard questions you can ask, but one question you never ask is, what's the relevance of your research? Because the speaker's embarrassed enough as it is. So fortunately, in our case, that's SQUIRP, and with the SFS, there's a lot of relevance to the research here, as I realized yesterday and from what I knew from before. So bear with me. So my outline is I'm going to give you a little bit of introduction about Archaea. Some of it may be familiar to many of you, but perhaps I'll give you a little bit of a different perspective. Archaea were not discovered that much that long ago. It's about 50 years ago. There was Scripps, I think, had a towed submersible. It found a temperature anomaly because hydrothermal vents had been hypothesized, but had not yet been confirmed. And then Woods Hole was part of an expedition where they had a submersible with a three-man crew. They were operating it for the U.S. Navy. And I think a towed submersible went down. Submersible went down. It found a temperature anomaly, and then it's got a photograph of hundreds of clams. And then Alvin went down a few days later, and they found all kinds of life. Before that, we thought photosynthesis was critical for life, that you couldn't have life without sunlight. So people were expecting if they found any life in the deep, dark ocean, it would be like a desert, but maybe there'd be a few clams living off the photosynthetic zone particles of life that filtered down. And they called it, they were looking for, they said, the $30,000 clam, because they thought they'd find just a few. Because they didn't expect life. I don't think they even had a biologist on that cruise. So they ended up with a whole lot of specimens. I think they ran out of formaldehyde, and then they were using vodka to preserve the samples, which I think is probably good for us, because if they had drunk the vodka, they may not have written the excellent research papers that came out. Keeps off and on its own. Sorry about that. And we've got 20,000 papers or more, that came out of there. So after that little bit of introduction, I want to talk about how we need to think about algae in lakes, which is a problem, as we've been focused on, not just by thinking about algae, but also the bacteria and the Archaea. And oftentimes, at least in the regulatory community, we kind of conflate all these terms. So we say microorganisms, or we say bacteria, but we forget that there are bacteria and Archaea. It's not just bacteria, and they play a very significant role. And then I'm going to talk about how, especially if you think about stormwater, which is high in organic carbon, nutrients, when we recharge the aquifers, it's a low oxygen environment, so you generate more methane, which is a much more potent greenhouse gas than CO2. So we are actually, stormwater management practices are generating greenhouse gases. So when we evaluate effectiveness of PMPs, which SQUIRP does, we should think about, what's the carbon dioxide equivalent, even in the construction, the concrete, the transport, et cetera. There's a buried cost, but there's also, as Professor Rajen showed us yesterday, there is a net carbon emission from stormwater pond. Now, they're small in size, but they're large in number. And the fluxes, as he showed, and I have a slide about his work, that the fluxes from stormwater ponds are much higher than from natural lakes. And then I'll talk about the bit of an aside, as I said, it's just one slide about how Archaea could help us deal perhaps with, say, carbon dioxide and oil and gas flares. Thank you. So Archaea, they are a different branch of life than bacteria. We can tell that because their cell walls are different, their DNA is different. So if you look at the origin of life, you start from the bottom, there's got to be a common ancestor because we share so much in common, right? All of life. So we do know there was a branch because, as I said, the cell walls are different. The DNA is different. And we, in fact, we come from eukaryotes, we're much closer to Archaea than to bacteria. So likely what happened, that's number 2 there, that bacterium fused within Archaea, and then we had the start of eukaryotes, where you have a nucleus. And in fact, I believe that a species of Archaea is known as the Asgardians. So there's tens of thousands of different kinds of Archaea. When we first found them, we thought they're extremophiles. They live only in high temperature or very low pH, but they are everywhere. They're even on our skin and in our gut. And then later on, we had one more fusing, which led to mitochondria. That's number 3. And then eventually, we probably had some chloroplasts merge with the eukaryotes, and then we got plants. So if you think about Archaea, they're quite different from bacteria. They were found in extreme environments. And I actually believe that we are the extremophiles. It's much easier for life to take advantage of high gradients. So when you've got high thermal gradients, you've got high pH gradients, life is energetically more favorable. So living at these low gradients is probably a much more extreme, takes more evolution and use of oxygen, to create the kind of life forms that we think is the norm. And we think of the high temperature, high pressure, low pH as the extremophiles, but perhaps it's the other way around. And if you think about Archaea, if you look at any water column, especially in coastal regions, they could be 10 to 50% of the microorganisms. And in sediments, many studies show they could be 90% of the organisms. In the subsurface, that can vary based on the geochemistry and many conditions. But we know there's 3 species on our skin. There's one in our gut. In fact, I saw a very recent paper that showed that children and adults, seniors have different levels of Archaea than adult humans. So we're not quite sure why that is. But the ones on our skin are probably oxidizing ammonia. That's what we suspect. So there's a whole lot that's unknown. But we do know that they have extremely vital roles. So why is this important? Because almost all anaerobic digestion, where we create methane from carbon sources, especially CO2, and in some cases from a hydrocarbon substrate, is done by Archaea, 98%. So in a wastewater plant, when they have an anaerobic digestion system, that's really Archaea that's doing the work. So they won't die when we chlorinate and kill the E. coli. So in the wastewater stream, which we then put into lakes like in Lake Elsinore, we really don't know what are the organisms other than, say, nitrogen and phosphorus that we seem most concerned about. What are we adding to the lake and what role are they playing? And they're playing a very critical role. So we need to think about it more as a system. And as I said, some of them will create methane from the CO2, and then some will, along with bacteria, convert the CO2 back to methane. So there's this whole interchange happening. And people are finding that even if the Archaea, 10% of the population, they may be responsible for 50% of the nitrogen fixation, and perhaps bacteria, the other 50%. So the other thing we have to realize is, as what was discovered at the hydrothermal event, that you can oxidize the sulfur and then reduce the CO2 to make hydrocarbons, right? So that's chemosynthesis. So then sulfur becomes a nutrient that's important. This is certainly not regulated. We don't worry about it, but we worry about nitrogen and phosphorus. But there's a whole range of organisms that will grow, and then they make carbon bioavailable. So then that makes other life forms possible. Then that helps with nitrogen fixation. So when we try to understand a lake system, the biogeochemical cycles of, say, carbon, nitrogen, sulfur, we can't avoid looking at sulfur because sulfur is one of the nutrients. Whereas when we think of nutrients, we are thinking nitrogen, phosphorus. I'm not going through everything that's on the slide because I'm hoping some of you may want copies. We can send you PDFs, and then you could you'll have more information than just me talking. So, thinking about algae in lakes, we know that if we have excess nutrients, then we get algal blooms, right? But when they die and they deplete oxygen, that's because of the bacteria and the archaea consuming that organic matter. So some of that CO2 and biomass will get converted to methane in the sediments, and then some of that will get converted to CO2. So there's all these interactions that are producing and consuming organic matter. We can't forget these. We have to think of the whole system. We can't just focus on the algae. And I'm just kind of reiterating that point. So here's a paper I found. There's actually a bunch of studies, but they're not all kind of collated yet, and very few for California that I found. So this was a global study. They were looking at organic carbon in groundwater. So you see the lower right hand, those are the countries they looked at. They had 9,000 odd global sources. And the figure above shows basically when it's hot and dry, you have less organic carbon in the groundwater. But when it's wet and mild, there's a lot of vegetation, then you'll have higher organic carbon. So basically, the groundwater quality, how much dissolved organic carbon is in the groundwater, will be a function of climate change, urbanization, the inorganic chemistry, the aquifer age. But the key point for us is that the dissolved organic carbon increases with urbanization. So when we have increasing land cover, we're going to have more carbon in the subsurface, which means more methane than would normally have happened where we would have had CO2. And methane is about 14, maybe, depends on how many years you're looking at and how you want to calculate it. But there are EPA calculators one can use, can range to 30 times the CO2 equivalent. So we know that if we have high dissolved oxygen, organic carbon, then we'll reduce the water quality, we'll increase treatment costs, and we'll worsen groundwater stress. And this is a paper that Professor Rajen actually approved my slide when I saw that he was a keynote speaker. I sent it to him. So he said, yeah, I think you captured my paper. But the point here was that they found that over time, the stormwater BMPs, the wet ponds were, yes, there was vegetation and they were capturing carbon, they were burying carbon, but the release of methane and CO2 was much higher. So they found between 20 to 200 grams of carbon per square meter per year was being captured, but almost 2,900 grams per square meter per year of carbon was being released. So as Rajen pointed out, these are small in size, but large in number. And I couldn't find a similar study for California. So I think this is work that needs to be done. This is from Florida. So go ahead. Thank you. And the point here is that the stormwater wet ponds can be net carbon sources, but they didn't look at stormwater recharge. I think the same thing works. When you recharge the aquifer, you're going to go into a low oxygen environment. That means you're going to have much more methane than CO2. So if you kind of capture that, here's a schematic. So if you think of the precipitation in the urban areas, paved lots, roads, you get all this storm drains bringing increased precipitation down a river. And then like on the Santa Ana, we have Prado Dam. We create recharge basins. But as I said, this is an overlooked issue that now we have increased methane. But if you think of pre-urban, it's likely more diffuse, shallower. And if you assume that the urban stormwater has, say, 10 milligrams per liter more organic carbon and higher levels of nitrogen and phosphorus, then we're basically recharging deep aquifers faster. And more than we would if there was non-urban rainfall. And as I said, the methane, excess methane is 30 times CO2. So on the Santa Ana River at Prouder Dam, approximately, I think, 55,000 acre-feet is being recharged at the moment. So if you consider that 50,000 acre-feet, that's like 60 billion liters, if you think 10 milligrams per liter excess. And I was looking for conversion factors of how much of that organic carbon would turn to methane. So again, I found studies from the East Coast for aquifers. I couldn't find a study for the Southwest or California, but conservatively, I think 0.2% of that carbon would convert to methane. Some would say maybe 0.4 to 0.6. And of course, when we extract that groundwater, all of that methane does get released. But that would calculate out to maybe 36 tons of CO2 per year. If you had 20 milligrams per liter and say 0.4, that would be 144 tons. So to put that in perspective, when we treat water and we transport the water, that's the real energy cost, right? Not of extraction. But people have done studies like SOPA in our region did a study on greenhouse gases from water extraction, treatment, transport, disposal. And they found that the extraction methane equivalent is like 1,000 tons. So if you look at this entire watershed, which would be 5 times what's going in at Prouder Dam, well, that becomes equivalent, right? So this excess methane from the recharge is equivalent to the methane from the extraction of groundwater. Now, I'm not arguing against recharge, don't get me wrong, because it's cheaper, less energy to extract at 50 feet than 500 feet. But perhaps we could think about how we do this. Like from Reisinger's work, I'd say you could think about the stormwater ponds being deeper and maybe less surface area, so you'd have less flux, you'd have more roll, maybe you have more diversity, so there's more interchange of the methane back to CO2 and from CO2 to methane, so you don't release as much fluxes. And here's a study, this is for the LA Basin. These folks were looking at where's the methane coming from. There's high methane in groundwater in the LA Basin. It's not from oil and gas fields. They did isotopic studies. They found as high as 150 milligrams per liter, but most of it was from biological origins. So when we extract this groundwater, clearly we're going to be releasing this methane. It's an overlooked source. So finally, this is my last slide. As I said, I was thinking about oil and gas fields. I did work in oil and gas as a process engineer. And oil and gas flares, they are high in sulfur. They have a composition very much like hydrothermal vents. Here's one of those. They're called black smokers that Atlantis and Alvin and others have found. But if you look at these oil and gas wells, it's hard to do much with that gas because it's sporadic, right? Suddenly there's a pump failure, a compressor failure. You got to release 1 million cubic feet. So you have to burn the H2S so that it's not toxic. But we release SO2, which makes acid rain. We also, 20% of oil and gas production goes into the atmosphere. Methane is a highly potent greenhouse gas. But you can imagine we could put into a tank with Archaea, and then they'd make complex hydrocarbons out of that. And then that could be a low-grade fuel. So I've got some folks who are kind of interested. I presented this concept, so we're trying to do some bench scale studies. I know at Virginia Tech, they're very interested in kind of following up on this. They have an Archaea lab. They also have H2S available. So, and finally, to conclude, Archaea are not the same as bacteria. They're critically important. If we want to address oxygen depletion, HABs, we should look at the entire system. And the higher organic carbon in stormwater, when we recharge into low oxygen layers, we're going to create more methane. This could be a significant source of greenhouse gas emissions. And there's perhaps a role for Archaea to capture carbon in oil and gas field flares. So thank you very much.
All right, I'm told we have time for one question.
Yes, so.
Do we have good capacity to actually measure Archaea where they occur to the same extent that we do for other microorganisms?
Yes, and I actually was very fortunate to attend a conference on Archaea quite recently. It was one of those Gordon research conferences. So all the participants presented. I had a poster session, had a poster there, and people were saying it's very difficult to actually grow and culture Archaea, not as easy as certain strains of bacteria, but there's just like we do with eDNA, it's called MAG. You take snippets of the DNA, put it into a bin, you have a model, you can figure out that these are unknown species or not. That's how we've discovered about 20 to 30,000 new species of Archaea. So, but not all of them are difficult to grow in the lab. There are people working on it. But as I said, it's very new. You'd be surprised, but if you did a search on Archaea, I'd say 50% of the papers are from China. They're studying it very actively. If you think about that also, we should put more emphasis on it. They're not the same as bacteria. So some Archaea have toxins against bacteria. So I'd say they have a symbiotic relationship, but there's clearly, they compete for resources. So we could use that to advantage, right? If we are thinking about algae blooms and the role of bacteria in oxygen depletion. It's not just bacteria. Once we realize bacteria and Archaea, maybe we could use that to control that oxygen depletion. Thank you. Thanks for pointing that out.
The same individual sent two questions. Well, the first one's a comment. It says sewage seems like a good food source for Archaea. And then their question is, do you know if there are any applications of Archaea-based technologies in reducing green emission or in reducing gas emissions from sewage?
I'd say that's an active field of research. I personally am not aware of it, but there is a database of people who find Archaea, who test for it. They put it online. So I've been looking at it, and Archaea are in wastewater effluents. Again, as I said, all the anaerobic digestion, all the methane being created in the wastewater plants is coming from Archaea. People now realize it's 98% Archaea, whereas earlier we thought it was bacteria doing the work. So I would say it's wide open. As far as I know, there isn't, but they are being used already. I mean, so I can perhaps I'm not clear about the question. We should discuss it later. But Archaea are doing the work of anaerobic digestion in wastewater treatment. But I would suspect that the E. coli being killed by the chlorine, because I asked a lot of experts, I said, well, what happens to Archaea? They said, oh, they probably just sail right through because they can handle the low pH and they can handle the oxidation.
Thank you, Raj.
Thanks, sir.
All right, so our next speaker is Emma Walker, who is coming in remotely. And Emma is going to talk about some slightly larger organisms, which BMI and Sierra Foothills streams.
Great. So hi, everyone. My name is Emma Walker. I'm the Benthic Macroinvertebrate Lab Manager for Sierra Streams Institute. And today I'm going to talk with you a little bit about visualizing trends in benthic macroinvertebrate communities in the Sierra Nevada foothills. So you can go ahead and move forward. Thank you. So for Sierra Streams Institute, we are a nonprofit organization dedicated to monitoring, researching, and restoring watersheds in the northern Sierra Nevada foothills. We are a community science-based organization, meaning we incorporate community volunteers into each of our programs, allowing community members to learn, engage, and interact with their local environment. Our programs include water quality monitoring and bioassessment, as well as education, outreach, forestry, and restoration. Our longest running program is water quality monitoring and bioassessment of Deer Creek. Okay, move forward. So the Deer Creek watershed is located in Nevada County. From its headwaters, it runs for 36 miles down an elevational gradient of about 3,500 feet before joining the South Fork of the Yuba River. And you can go ahead and just move forward a couple of slides. Basically just highlighting here that along its course, it flows through 2 dams, Scotts Flat and Lake Wildwood, as well as the town of Nevada City, which impact water quality in a number of ways, some of which we'll explore a little bit today. We began monitoring Deer Creek in 2000. We have 18 sites which are monitored monthly for chemical and physical parameters of water quality. And then a subset of these sites are monitored for benthic macroinvertebrates. Moving forward. Yeah. So all of our benthic macroinvertebrate samples are collected and identified to family level taxonomy by community volunteers. Using a community science approach allows us to build capacity for our program and connect volunteers to their environment and create, and collect a data set that allows us to better understand our watershed. Using this data set and a number of visualization trends, tools, we're going to explore how our watershed changes spatially, in addition to how it changes in response to changes in environmental conditions like flow. Using visualization tools like the ones I'm going to show later, we can employ our kind of coarser taxonomic resolution, our family level data, and present our data in a way that is easily accessible to our community and our partners. At family level data, we can use a number of metrics. The first being percent EPT or percent Ephemeroptera, Plecoptera, and Trichoptera. These are orders which are known to be more sensitive to changes in environmental conditions. We can also look at EPT family richness and diversity. And we can also look at the overall community. So that includes all orders, not just those within Ephemeroptera, Plecoptera, and Trichoptera. And lastly, we can look at functional feeding groups. Functional feeding groups tell us not only the type of food consumed by an organism, but the behavioral morphological traits used in consumption, and can give us a little bit of an insight into the trophic structure. So using all these metrics and looking at previous research, we expected to find higher levels of EPT, higher levels of richness and diversity in our upper watershed sites, as well as when we have wetter conditions. Conversely, we expect to find lower levels of percent EPT, lower levels of diversity and richness in our lower watershed or in drier conditions. So let's dive into that. So we collect samples 2 times per year during June peak flow or October base flow conditions following the swamp protocol. Samples are then identified to family level taxonomy by community volunteers under guidance from our lead entomologists and taxonomists. For today's analysis, we focused on samples collected between 2003 and 2015 and confined the analysis to 11 sites that had more than or equal to 10 years worth of data. And finally, for when we wanted to look at our flow, we used data collected from the USGS Smartsville gauge, which I'll get into a little more in just a moment. So the sites you see highlighted in red are those included in our study. They include sites along the main stem as well as within 2 tributaries. So here we have a graph from the National Integrated Drought Information System, basically showing our analysis period, so 2003 to 2015, and this shows the overall precipitation for the state of California. Within this time period, we observed both periods of sort of wetter conditions, that blue color, and drier conditions, that red color. But we really wanted to get a more localized view of our watershed, so we looked at the USGS Smartsville flow gauge. So that flow gauge is indicated by that blue mark at the very base of our watershed. Because it is below both dams and diversions, it really only serves as a proxy for the flow conditions throughout our watershed. And knowing it was a proxy, we used it to calculate historical percentiles and broadly categorize our flow conditions. So historical percentiles were calculated. Sorry?
Oh,
sorry,
you're back. You cut out for a few minutes.
Am I cutting out?
Seems to be okay now.
Okay, great. Sorry. Great, to keep going, historical percentiles were calculated for the 6 months prior to our collection. So for our June collections, we looked at the January through June period, and for October, we looked at May through October. We wanted to look at the 6 months prior to our peak flow and base flow, because we found that our BMI communities, our benthic macroinvertebrate communities, were most significantly impacted by and responsive to the period 6 months prior. So if the graph on the right showing sort of our peak flow conditions, which overall higher flows, while that graph on the left are base flow conditions and overall lower flows, but definitely much more flashier. So once we had our historical percentiles, we classified them into flow categories based on the USGS Water Watch. So drought, anything that fell below 10th, dry, anything below the 25th, wet above the 75th percentile, and flood above the 90th percentile. Once we knew what these broad flow thresholds were, we then compared these thresholds to the flows of the 6 months preceding our actual sampling events. Go forward. And we found that our samples were collected during flow conditions which fell within each of the flow categories. So dry, drought, average, wet, and flood. So now that I've given you a little bit of background into how we did our sampling, how we looked at our flows, let's go ahead and get into some of these visualizations. So as we began this investigation, we first wanted to see how these communities varied along our elevational gradient. Go ahead. We created what we call heat maps to explore the spatial distribution of percent EPT at each site over time. So each cell here indicates one sampling event, while the color corresponds to the percent EPT, with green indicating a higher percentage and red indicating a lower percentage. These sites are our main stem sites and are ordered as they descend down the watershed. With this tool, we begin to see a lower percent EPT in our lower watershed site, particularly during our fall or base flow conditions. Go ahead. We use the same tool to look at overall EPT richness, and we see a lower EPT richness in our lower watershed sites, consistent both in our spring and fall sampling periods. Please move forward. We also use similar visualization tools to conceptualize the distribution of our functional feeding groups. In our upper watershed, we do see a higher percentage of shredders. Well, in our lower watershed, we see a higher proportion of collector filters, indicated in gray. And this is consistent with the idea of a river continuum and the differences in food availability along our watershed. So please go ahead. So using those preliminary tools, we begin to pick up on a spatial variation in our BMI communities. To further investigate how these communities respond to change over time, we looked at ordinations. Ordinations plot samples in clusters based on their similarity. The more similar community composition is to one another, the closer 2 points will be on the graph. Go ahead. We can then overlay grouping variables to identify patterns of similarity within the clusters. For example, when we group by site, we see samples collected from the same site clustering together. Move forward. And so grouping in this way allows us to begin to see some differences between our upper watershed and our lower watershed sites, with the upper watershed sites clustering towards the left of that plot and the lower watershed sites clustering towards the right. Moving forward with analysis, I did keep all the other ordinations within site to control for site-specific differences, like changes in habitat and riparian cover. So the ordination you see now comes from site 2, one of our upper watershed sites. So the samples were plotted and then grouped by their flow category. And when they're plotted in this way, we begin to see a difference between communities collected during dry conditions and those collected during wet conditions. We can then overlay our metrics as vectors. Please go ahead. And we see that wet conditions are significantly correlated with higher percent EPT, higher EPT community, higher EPT richness and diversity, and higher community richness and diversity. And so what I'm showing you is just one plot, but this pattern was true for all of the sites that you see in red on the map, and is fairly consistent with what we were expecting. Please move forward. We then did a similar analysis for our lower watershed sites, again, first plotting our samples and then grouping them by their flow category. Go ahead. However, this time when we overlay our metrics, we see that it's in fact drier conditions that are more correlated with higher EPT richness and diversity and higher diversity and richness. And now the ordination you see here comes from our site 6, but this pattern was observed at 2 of our lower watershed sites, and varies from what we were expecting. Let's go ahead. So what mechanisms are driving different responses in our upper and lower watershed sites? Well, using our initial spatial visualizations, we began to tease apart those differences between the upper and lower watershed communities. And so it's possible that these sites have different communities, which have different responses to changes in flow conditions. And just to kind of look a little further into this, we dove into some of the families driving these trends. Go ahead. So first, looking at our upper watershed sites, seeing which families were significantly correlated with wet conditions. We see a number of families which are known to be found in colder, higher elevation streams, and more sensitive to changes in environmental conditions. Go ahead. And looking at those communities associated with drier conditions in our lower watershed, we see families that are generally adapted to warmer, low flow habitats, more stressed environments. And so while there was a little bit of overlap, we do recognize that there is some variation within these families that could be contributing to that. Go ahead. And again, just to further look into this, we wanted to see how our temperature varies along our entire watershed. So we found that on average, our lower watershed is higher than our upper watershed. And that was consistent when we separated out by season between wet and dry years. Consistently, our lower watershed was much warmer. So our lower watershed, it's warmer, it's below dams and diversions, and all these things can contribute to the different communities that we believe we're finding. So what's our major takeaways? Well, flow matters, but not always in the way that we expect. So our upper watershed, we found that increased flow was associated with increased EPT diversity and richness. While on our lower watershed, we found it was in fact decreased flow that was associated with EPT, with increased EPT diversity and richness. And I think this analysis really highlights site-specific nuances, and so it can be really difficult to make broad, generalized claims about community responses when we find such variation along a watershed, even in a watershed that's the size of Deer Creek. As you know, California continues to change with climate change. We expect to see more extremes, more droughts, more floods, and how our communities respond to those changes can vary along the watershed. So go ahead. So we want to continue with our community science-driven approach to bioassessment, but we are hoping sort of to modify our program moving forward to help better our analyses and better understand how they're changing, how our watershed is changing. So the first of those, we are shifting to a higher taxonomic resolution. And so while family level data is really can be useful for broadly categorizing structure, there are variation at the genus and species level in terms of their tolerance values, their functional feeding groups sometimes. So we are moving forward with higher level of taxonomy. We also hope to increase the resolution of our flow data so we can get a better idea of what our flow is like throughout our watershed, as well as potentially diving into some of the cumulative impacts of like prolonged drought. We are continuing with our program, and I hope to expand this analysis to include more recent years. So we do have samples collected from 2021 and 2022, two of the driest years in California, as well as samples collected from this past summer and fall. So we'll look and see how some of the wettest conditions now impact our communities. And the last thing that we're hoping to do is to expand into sentinel sites. And by this, we just mean sites that have really minimal impact from things like development. And so we can get a clearer picture of how flow impacts the community without sort of that other interference. And so we can move forward to the last one. So I just want to say thank you so much for taking the time to listen to this presentation. And also a big thank you to the volunteers who helped us do this and collect this data. So thank you so much.
We have plenty of time for questions.
Hi, I'm Ray McNish from Cal State Bakersfield. Thanks so much for your talk. It was really interesting. I have two follow-up questions. So if I'm correct interpretation, your NMDS ordination is focused on the taxonomy of the community. I'm just wondering, as you're continuing into the future to explore your data, if you thought about swapping out taxonomy and also exploring the functional aspects of your community, because you can have taxonomic changes, but the functional aspect of community might still be the same. So I'm just curious about that. And then also, if you're considering doing follow-up statistical analyses with the NMDS ordination, like indicator species for like which taxa or traits are indicative of your different sites or habitats that you're focusing on.
Yeah, absolutely. So just in terms of moving forward, we have a lot more data that didn't get incorporated into this analysis. A lot of physical habitat data. And you're right, there's other ways to examine these communities, looking at like what functional feeding groups are present. And I think would be absolutely fantastic to explore those ways. As I also talked about a little bit earlier, we have a water quality monitoring program. So we do have a lot of data about the chemical and physical parameters. So not just what the biology is like there, but things like temperature, dissolved oxygen, pH. So I think it would be great to continue this analysis, incorporating all those different pieces of data. But it's just not something that we have done yet. Does that answer your question, or did you have something else?
Yes.
Other questions? Anything online? All right. Thank you, Emma.
Thank you so much.
All right. So our last speaker of the session is going to be Melissa von Mayrhauser. Did I say that right? Yes. And so we're going to go even bigger on the organisms. We're going to be talking about birds and habitat restoration in effluent-influenced streams.
Thank you, Chad. How's everyone doing this morning? Good morning. Yes, as Chad mentioned, my name is Melissa von Mayrhauser, and I'm a PhD candidate at UC Berkeley. I'm really delighted to have the chance to speak with you all this morning about some research I've been working on with some collaborators about understanding the associations between flows and riverine birds to inform the restoration of effluent-influenced streams in coastal Southern California. Wanted to begin with a question for all of you, which is, what is something these two bird species may have in common? Is anyone a birder here? Okay. Maybe by the end of this presentation, you'll feel like, oh, birds are great. So something maybe that could come to mind is that these birds, the song sparrow and the black-necked stilt, are both insectivorous. They're both residents of Southern California, and they're also known for their unique vocal stylings. Another possible answer is that these birds are both dependent on stream flows. So the song sparrow, for example, lives in shrubby habitats close to fresh water, and it nests on a lot of riparian vegetation. The black-necked stilt nests on a lot of emergent vegetation and sometimes even on mud piles in the middle of the LA River in a concrete-lined section. And it also forages in these open water habitats. So birds like the wonderful black-necked stilt and the song sparrow, as well as the yellow warbler here, are examples of how birds are excellent bioindicators, but they are not typically included as key indicators in a lot of stream monitoring efforts. And this is despite the fact that they exhibit this wide range of sensitivity to environmental change. There's a low level of lab analysis needed to understand them. There are many people with ID expertise, and they're very important to a lot of people. A few examples of this. Has anyone used the eBird app? Maybe not. Yes, I see some nods. Okay, yes. So the Cornell Lab of Ornithology has this app where it's possible for anyone to become a user and upload checklists showing observations of birds. So in LA County alone, there are over 20,000 users of this app, and they've completed over 500,000 checklists. So those are a lot of data that are available to us online. And here we have the great blue heron that's been a symbol of a lot of LA River revitalization efforts. And birds are also so culturally important. And one example is the prominence of a bird in this Tongva creation story. So we are thinking about birds in the context of these two rivers in Southern California, the Santa Clara River, also known as the Utam River in Chumash, and the Los Angeles River, also known as Paime Pahait in Tongva or Orit in Tataviam. And these are really interesting rivers to compare. The LA River has this history. It's known for being a river that's been so channelized and mostly covered in concrete, although not entirely. Whereas the Santa Clara River is known for being one of the last remaining more ecologically intact rivers in Southern California in this area. So we're comparing these two rivers, and we can see some of the differences between them that I just mentioned here with these satellite images. And we see the extent of some of the in-stream riparian and upland changes along the LA River here, compared to the overall more intact Santa Clara River ecologically. In the case of both of these rivers, treated wastewater or effluent has flowed into them for the last several decades, and it's homogenized their flow regimes in some reaches. But right now there are planning efforts happening around this desire to use more of this water potentially for more local water supply needs in an era of climate change. So perhaps using it instead for groundwater recharge, local irrigation, or even drinking water. So we're thinking through these trade-offs now. Is it better to put more of this water into our rivers or use it for other needs? And we are moving in the direction of discharging less of it into these rivers. So with that context in mind, we are asking, how are bird communities associated with patterns of effluent and flow in these two watersheds? We've established these 14 sites that you see here in yellow or gold. And at each of these sites, we have 3 transects. And for the LA River, you can see we have 3 sites that are upstream of effluent influence and 3 sites that are downstream of effluent influence that are effluent dominated, especially because, like I mentioned, so much of the LA River is covered in concrete. So the effluent is not getting into the groundwater in the same way that it is on the Santa Clara, which is not channelized or concretized in the same way as the LA River. So we have some sites that are unaffected, and then we have quite a few sites that have varying degrees of effluent influence, maybe depending on the time of year and the season. And so we have a lot of subsurface flow influence in the Santa Clara River that's affecting riparian vegetation that we don't see on the LA River, excuse me. So we've been visiting these sites over 3 seasons and 2 years. And here we've been conducting point counts. So when we go out to do point counts, we go to 2 sites per morning. within 3 hours of sunrise. And when we go, we're recording all the bird species that we see and hear for a 10-minute period. And we use 2 recording devices. So the audio recorder you see in the middle, as well as the Merlin app, which also comes from the Cornell Lab of Ornithology. And we use a long lens camera. So this allows us to have a lot of information to go back to after we finish in the field. And at the same time, we conduct vegetation surveys and aquatic and terrestrial invertebrate monitoring. I'll get to those at a future presentation. And at this point, I'd really like to give a shout out to my collaborator, Tevin Schmidt from the Wishtoyo Chumash Foundation. And he's been an invaluable field partner. Thank you so much to Tevin. After collecting all of these data, we're going back next month to continue on with some surveys. But as we've collected all of these data, we've assigned the bird species that we've found traits that allow us to study the association between environmental change, in this case flow, and bird community structure. So for the purpose of this presentation, we're focusing on habitat dependency. And so this is the extent to which birds are dependent on habitats for foraging and breeding. Here are some examples of how we have divided these birds into these different trait categories. So you may recognize the black-necked stilt from earlier, an open water dependent species. We have the song sparrow in the riparian dependent category, the California quail in the upland dependent category, and our friend the house finch in the generalist category, meaning that birds in this category are not particularly dependent on one type of habitat for their breeding and foraging needs. So with all of this in mind, we predict that reductions in effluent discharges will most affect open water dependent and riparian dependent bird species. And we think this will particularly be the case for resident species that are vulnerable to flow changes all year round and not just for part of the year. And we think that this prediction is consistent with the Square Belly River study that has looked at how reductions in effluent would affect, for instance, willow and green algae, which would then affect these birds that we see here as well. So in terms of what we've seen so far, we found 136 species across our surveys thus far. And on the left, we see the most common LA River birds, and on the right, the most common Santa Clara River birds. So you may recognize the house finch, that generalist species in both of those. On the LA River side as well, we see 2 open water dependent species, the black phoebe and the mallard. On the Santa Clara side, we also see a California towhee generalist species, but notably over here, the species kind of tipping its head back and singing is the Buick's wren, which is a riparian dependent species. So we're starting to see some differences even in looking the 3 most common species across these 2 watersheds in the way that they're using their habitats. So we wanted to give you a sense before we dive deeper into the traits of some summary statistics with what we've found so far. So we have richness on the left and abundance on the right. And the LA River, both for this slide and future slides, is in purple, and the Santa Clara River is in orange. So just generally, we found higher richness values for our bird species along the Santa Clara compared to the LA River, and higher abundances for the LA River, compared to the Santa Clara River. But we can see more of a spread in the data across our sites for the LA River compared to the Santa Clara River. And this is somewhat consistent with what we're finding for this NMDS, our good friend, the NMDS that we've seen a few times in some presentations, the non-metric multidimensional scaling. And as we heard, for instance, from Emma before this, the closer in space the points are, the more similar they are to each other. And this is looking at an NMDS for the traits that we mentioned about habitat dependency, with the LA River again in purple and the Santa Clara River in orange. So something that we wanted to give you as a takeaway message from what we're seeing here is that you may note that the Santa Clara River sites trait data are much more clustered together than what we're seeing for the LA River, with a much greater spread. So regardless of whether we're upstream or downstream of effluent or in a dry or wetted site for the Santa Clara, we're seeing that clustering. Whereas generally, as we're moving from upstream to downstream on the LA River, we're seeing more variation. So that last NMDS was helping us to think about the general sort of dissimilarity in the data and the traits that we're seeing, but it brings up questions of why is this happening? So to further illustrate this, thought we would provide you with these pie charts that show you the proportion of habitat traits per site. And you'll note there's only one pie chart per site because we've taken an average for ease of visualization. Otherwise, we'd be throwing a lot of pie charts in right now. And we see, we're starting to see some differences between the LA River sites here and the Santa Clara River sites. But before I get to those, I wanted to provide some context. So for the Santa Clara River sites, I've circled them all in green here. They're all unchannelized. And so is LA River 3. So we can start to see maybe some similarities here between LA River 3, for instance, and Santa Clara River 4. LA River 4 here is in this dashed line because it has a soft bottom and concrete banks. And these descriptions compare to what we see or contrast with what we see for LA River 1 and 2 and 5 and 6. And we've circled them in a gray line to indicate that they're fully concrete lined. All right, so we can use this information to start to interpret what we would expect from effluent reductions for these sites. And here we see the solid blue line for sites that are effluent dominated, and we see the dashed line for sites that have some effluent influence to a varying degree. So for the LA River sites, we see just how many open water dependent species they are supporting. So in terms of all of the blue that we're seeing here, compared to the Santa Clara River sites that are generally supporting more riparian and upland dependent species. So a reduction in effluent on the LA River could have a more immediate impact on these open water dependent species that are there for that water. Whereas along the Santa Clara River, we would expect more impacts on the riparian dependent bird species, especially because so much of the riparian vegetation is being fed by that effluent subsurface flow that I mentioned to you earlier. So a takeaway message is that changing effluent discharges will affect the two rivers and their bird communities in different ways. And so this has implications for how managers are thinking about reducing effluent in different types of streams, looking at, for instance, how the characteristics of a stream like the LA River with the concrete lined sites could be so different from managing effluent reductions in a less channelized stream system like the Santa Clara. And so we're going to continue to work on our data analysis in this area, bringing in our invertebrate data and vegetation data as well. But in the meantime, we are also thinking about this as a social question. So we're thinking about reductions in flows as something that not only affects ecosystems, but also has social implications. And so as part of this, I'm actually going to be conducting surveys or interviews with people who work on the Los Angeles and Santa Clara rivers, asking this question. So how do people from government agencies, tribes, and community groups define their priorities for flow-based restoration along the Los Angeles and Santa Clara rivers? So if you work along one of these rivers, or you're interested in discussing this question further, I would encourage you to reach out to me, and I plan to start conducting interviews in collaboration with a social scientist from UC Berkeley in spring 2024. All right, and I'd like to acknowledge all of my collaborators, friends, colleagues, family, as well as funders. And thank you so much for all of your time.
Plenty of time for questions.
Great talk, first of all. And I know you just started on this research, kind of going through it. But the slide you had with the pie charts, it looks like you kind of have two things going on there, both the channelization of the stream as well as the effluent influence. Are you going to be digging into that a little bit more like statistically and probably with your other data? Because right now it seems a little bit too confounding to really tell.
Yes. Yeah. Yeah, so we're collecting our last round of data this December, next month. And then our next step will be to do a Permanova test and start to look at the nuances of what we're seeing here. This was definitely a simplification for the presentation. And I think it's really interesting to look at how, like, a lot of the sites along the Santa Clara River, sometimes we see that they have flows coming from effluent, sometimes not, sometimes it's subsurface flow. So I think it's important to take it to the next level when it comes to sort of thinking about all of these connections and start to do statistical tests that can help us look at the deeper meaning of, like, the relationship between flow and the bird community composition.
Fun study. Couple of things. A lot of the restorations that's been happening in urban environments along riparian areas, they've shown an increase in urban birds, not riparian birds. And this is great because it's going to help kind of dial in something that's been missing for a long time. And then locally, maybe 10, 15 years ago, Dick Zimbel did work when he was with Orange County on bats. And that may be a good correlation as well, because he was looking at water quality. And, you know, they share our same corridor, same space, a little different niche, but there's other stuff out there that might help you.
Eric.
Any other question?
Thank you very much. Yeah, it was a great presentation. It's interesting to see how you have more open water dependent birds than like in LA than the Santa Clara River. But I'm curious, when you go to your box plots, you had more variability in your LA River. Is that because it's more open water dependent birds there? And were those captured from just one time interval, or was that like over multiple periods? And I guess I also just curious with like future research, would it be possible to then measure discharges temporally just to have some sort of time series analysis? Or what I guess might be some cool next steps?
Thank you for those questions. I'll go back to this slide that you're referencing. And yes, for this presentation, it's an amalgamation of what we were seeing across the different sampling outings. And so we have, we wanted to just show the overall spread in the data. And I like your question about, like, why are we seeing more of a spread for the LA River sites? And I think these sites are so distinct from each other in a way that the Santa Clara River sites are not. So I think the Santa Clara River sites share a lot more overall compared to the LA River, where we have one site that's soft bottom but has hard banks. We have another site that's fully soft bottom with soft banks. We have concrete line sites that have urban runoff upstream and then effluent downstream. So I think we're seeing some variation that comes out of just how distinctive these sites can be from each other and how much we've engineered them in different ways that we don't see with the Santa Clara. And yes, we're thinking a lot about flow data. And I think for a future talk, I'd love to bring in a time series of what we see with USGS flow gauges along these streams, as well as flow data coming out of the wastewater treatment plants and how that's changed over time as well, and how it might relate to what we're seeing. So I think there are a lot of possibilities. Or using satellite imagery to look at water presence and absence. So I hope in future talks to kind of bring in those links a little bit more with the flow data, as well as the bird composition data, and maybe also throw in some vegetation invertebrates. Yes.
Thank you. Great talk, Melissa. I have two quick questions. First one is awesome. You're working with tribes and the Santa Ana River and LA River. Is the riparian area, at least particularly in Santa Ana River, is it a general gathering place, or are tribes using it in the riparian area for gathering? Is that an activity that has been ongoing increase that you know of? And then lastly, you did mention the eBird app and all of the citizen science data that's been collected. And I think your data was based on your guys' point counts, but are you looking at the citizen science data that's been collected over time? Does that kind of match with the patterns of what you guys are seeing for those sites that you were including in your study?
Yes, so I think that the questions about flow and tribal uses are so critical, and anecdotally, I've already seen that is occurring in terms of there are many people who are relying on many of these sites for cultural uses, where if certain types of vegetation were not there because maybe there's a reduction in flow, that could affect, like for instance, traditional basket weaving practices. And so with the social science study that I mentioned, I think it will be really critical to speak with tribal members and to hear a range of perspectives about how a change in flow might affect traditional cultural uses. So that's at the front of my mind as I go into those surveys, and I hope to learn more. And then your second question, I think there's so much citizen science data that's available, especially through eBird. And with those over 500,000 checklists, complete checklists even. So, and it's really interesting. You can also see who created the checklists, and there are some really incredible birders and professional, like ornithologists, who use that app as well. And there's so much data that we can glean from those checklists, even like who's creating them, how many checklists do we have in a certain site. Was there maybe a survey that was conducted on a similar day to the one I did? So we've been looking into those, and I think the data looks similar to what we're seeing. Although if we were comparing, we would just want to constrain what they're seeing to a 10-minute interval, which we could do. And we'd want to look at who is collecting the data and that sort of thing. But I think there are so many more possibilities given the amount of bird data that is available.
Great presentation. I was wondering if this could be adapted into like a statewide program, or do you think this is more appropriate for like a regional scale?
Yes. I think it would be great to see this as a statewide protocol. And for all of the reasons that we were mentioning back at the beginning about how great birds are as bioindicators, and especially given how important they are to so many people, but then also, yes, so many people with ID expertise who would be able to contribute. And just, it's been incredible conducting these surveys as well, because I can go out into the field, collect all the data, and then go home and I'm done. I mean, maybe I need to review a recording, but I'm not going into the lab to do extensive review. So there's a quick turnaround time, and they can tell us so much about how environments are changing over time, just with this wide range of sensitivities that they have. Yes, there's such incredible traits that they have beyond the one that I discussed today about habitat dependency, like their trophic niche, or whether they're like the rate to which they disperse. So resident versus migratory. So many different aspects to consider that could be integrated into a statewide protocol.
Yeah, one more question. I don't know who was first. You're winning the most questions asked prize.
I was wondering if you've done any analysis on listed species and how they might be utilizing, like, between the two rivers or different points within the two rivers.
Yes, that's a really great question. We're starting to think about sort of more of the characteristics of the species as well in terms of their conservation concern. So we've been starting to map out the species that we consider to be low conservation concern compared to ones that are sort of where the populations are declining or we're concerned, or even where the species may be threatened or endangered. And we've definitely seen more of those species along the Santa Clara River compared to the Yelly River. But we also see some important, more rare migratory shorebird, birds along the Yelly River as well. So I think that's a really important additional dimension to consider, and how a reduction in flows, for example, could affect especially endangered or threatened species, like the least-belled vireo, for example.
All right, thank you so much.